Hi, my name is Carrie Stephen, and I will be presenting today about a paper that talked about the metabolism of alternative sweeteners and how the microbiome can play into that. You have the citation at the top showing my paper. So, the scientific question or hypothesis that the authors were trying to convey and study in this paper was the impact, if any, of 4 different commercial non-nutritive sweeteners, or abbreviated NNS, throughout my presentation, and what impact they have on the human metabolic health and the microbiome. The researchers proposed that these non-nutritive sweeteners may modulate the gut microbiome and cause variable and individualized metabolic responses in the host consuming them. And they thought this because there were some previous studies showing that these NNSs sometimes impair glycemic responses, but without pinning down the mechanisms behind the impairment. And so they were hypothesizing that the microbiome is part of the mechanism behind this. So to start, let's talk about what these non-nutritive sweeteners are. In this study, they looked at these 4 that I'm showing you here. So aspartame, which is a common ingredient found in a lot of diet and sugar-free products like sodas, yogurts, gum. Saccharin, which is common brand is Sweet and Low. There's other brands out there too, but that's one you've probably seen. And sucralose, which the main brand there is Splenda. And then stevia, which is commonly Truvia, is a brand I see a lot. So these sweeteners have been touted by many, including a lot of healthcare professionals, as sugar replacements because they don't contain any calories. That's why they're called non-nutritive. And because of this, they're presumed to be metabolically inert and not elicit a glycemic response that's normally associated with sugar digestion. So many doctors and dietitians recommend swapping sugar for these replacements as a strategy to help fight against obesity, hyperglycemia, type 2 diabetes, and cardiovascular disease. Consumption of these sugars is very common. I'm sure many of you are probably familiar with some of these products. In a survey conducted between 2009 and 2011, about 25% of children and about 41% of adults in the US reported consuming 1 or more of these non-nutritive sweeteners. And I'm guessing the number has just risen since then. Another important factor to consider when we talk about this paper is that we are in the midst of a global health epidemic. Obesity has been on the rise in the past 4 decades, as you can see in this figure I have here. There's been a steady increase in the mean BMI around most countries and regions around the world. One study that I read estimated trends from 1975 to 2016 in over 200 countries and territories, and they found the prevalence of obesity was more than 30% in some countries in 2016, including Nauru, the Cook Islands, and Palau. And obesity rate was around 20% in some other countries like the Caribbean, North Africa, and the United States. And some studies vary on those numbers. I found another one that said the prevalence of obesity in adults in the US was more like 45% in certain subgroups. So obesity is definitely a big problem. And we all know that obesity is considered a chronic disease, and epidemiologic studies have revealed associations between high BMI and a large range of other chronic diseases like non-alcoholic fatty liver, cardiovascular disease, diabetes mellitus, several malignancies, musculoskeletal disorders, chronic kidney diseases, and mental disorders, which consequently are going to negatively affect a subject's quality of life and raise healthcare costs. And being overweight in childhood also has adverse psychosocial consequences and lowers educational attainment as well. So with this rise in the obesity and the rise of these non-nutritive sweeteners being touted as alternative options for sugar, you can see why us studying the effects of these sugars is very important. Another reason that it's important that we continue to study these non-nutritive sweeteners is that there's a lot of mixed results in studies that are previously out there. So there's some that suggest that these non-nutritive sweeteners do improve metabolic markers. There's some that are neutral, and some suggest that these sweeteners even contribute to obesity and diabetes in some contexts. But the problem with a lot of the studies showing negative impacts are that they're observational. So it's kind of difficult to determine whether their findings are due to reverse causality, such as, are those sweeteners causing the weight gain and hyperglycemia, or are people that have these conditions just more likely to be consuming these non-nutritive sweeteners? Also, a lot of these studies have variability in what outcomes they're looking for and their methodology, so it's kind of hard to compare them and interpret their results. In addition to human studies, there's been a lot of animal studies showing similar mixed results, some positive, some negative, some in between. Most of the animal studies show adverse effects to metabolic health, though. So the researchers for this study propose and end up showing some really compelling evidence that the microbiome might have something to do with the variability in results seen across these studies. Everyone's microbiome is unique, so that leads to very personalized responses to diet and treatments. And in regards to non-nutritive sweeteners specifically and their effects on the microbiome, while there haven't been too many studies, there have been some previous studies that show that these sweeteners affect microbial growth in culture, that they can modulate the gut microbiome in animals, and that those modulated microbiomes can cause glucose intolerance in germ-free mice. And there have actually been a few human studies on the microbiome and these non-nutritive sweeteners, but the researchers state that some of those previous studies didn't screen their participants for previous consumption of the non-nutritive sweeteners, which in my mind kind of invalidates their results a little bit if the microbiomes have already been affected previously before the study by these sweeteners. So the researchers felt strongly about testing that factor. And then a lot of the previous studies also used 16S rRNA sequencing or quantitative PCR of very targeted microbes, which doesn't allow for super species level specifics, which the researchers of this study wanted to get into more detail about. So let's get into their methods a little bit. You can see a figure here that shows an overview of their study design and plan for collecting a lot of different sample types. It was a randomized, controlled interventional trial with 4 intervention arms, one for each of the non-nutritive sweeteners that you can see colored in the top here, and then 2 control groups. And in total, they enrolled 120 participants, 20 in each group that completed the trial. And notably, those participants were all people who had not consumed any of the non-nutritive sweeteners over the past 6 months before the trial. And the researchers did a very detailed food survey asking all of them about consumption of very particular products that were on the market in the area to be sure that they got people that hadn't been exposed recently. And as you can see in the figure, this trial took place across a 4-week time period with a variety of a lot of different types of tests collected throughout. What I want you to mainly notice is that during the first week, we have baseline measurements of all of the different parameters. We have 2 weeks that say during supplementation, so they were getting these sugars or the control for 2 weeks with still more samples being collected throughout. And then there was a follow-up week recording and collecting a lot of follow-up samples for every test. We're going to mainly focus on results from the stool metagenomics, some of the plasma metabolomics, and glucose tolerance testing. And we're also going to talk a little bit about the transplant to germ-free mice, those results. I also wanted to point out a little bit more about the control groups and how the supplementation worked. This was not a blinded study because they believed that all of these sweeteners have very distinct flavors, so they just used commercial packets that they had to dissolve in water and drink several times a day. Each of these sweeteners, the commercial versions though, have a little bit of what they call a glucose vehicle in them. So they had to have a control group that was just receiving a little bit of that glucose vehicle, and then a control group that was receiving no supplementation at all. I also wanted to point out that researchers were able to keep really good track of compliance throughout the study protocol because the participants all had phone apps that they had to record every time they took their supplementation of the sweetener or control, as well as recording food and activity logs throughout the entire time. And so the researchers had instant access to all of this, and they were able to follow up and give calls to people who weren't adhering to protocol and keep them on track. They also had no significant differences in the food and activity logs of all of the different groups. So the results obtained can be more confidently attributed to the supplementation of these different sugars. So as for their sequencing methods, as I mentioned before, the researchers of this study thought that a weakness of some previous studies was their utilization of 16S rRNA sequencing. And so for this study, they performed shotgun metagenomic sequencing, which is the sequencing of many genes and genomes from members of a microbiota, not just part of a single marker gene like the 16S rRNA. The technology they used was the Illumina NextSeq platform with a read length of 75 base pairs single end, which I think was a really great choice for this study because that method has high quality and low cost output and a very massive throughput. So that's kind of the main highlight there, because of just the sheer number of samples that they were collecting throughout a longer time period, several weeks. For over 100 volunteers, they needed to sequence a lot of different samples. They also sequenced samples from the mice that had fecal microbiome transplants. So the Illumina platform was a good choice in order to analyze the large amounts of data that they needed to. And then for the analysis of the sequencing that they obtained, the sequences were QC trimmed using a program called Trimomatic. And then in order to classify into taxonomic groups, they used a program called Kraken2, and then a program called Bracken was used to estimate the actual abundances of all of those genus and species. They then assigned functional annotations to genes using several different analytic pipelines, and they grouped them into KEGG orthologs and calculated the abundances of those orthologs. So if you recall from genetics classes and things, orthologs are defined as genes in different species that have evolved through speciation events only. So generally they're considered to have the same biological function but in different species. And in many cases, I just wanted to point out that grouping genes this way is really helpful and it provides a lot of good information. But I wanted to note that the only really accurate way for determining the function of a gene or protein is by biochemical and structural studies. So there is a possibility of error and misidentifying genes in this way if it hasn't been previously tested. And then lastly, they assigned these genes that now are grouped by function to different metabolic pathways. And they used a program called Empanada in Python for that. So getting into the results of the study, we're first going to talk about the results of the glucose tolerance testing. So this figure is showing those results with each individual bar graph representing one of the supplementation groups or the control groups. And glucose tolerance testing, just how it works, is that it's a way to assess a person's metabolic response to a glucose load. So in this case, each participant was instructed to drink 50 grams of glucose after a set period of fasting, and then their glucose levels were recorded continuously over the next 2 hours. They were all wearing continuous glucose monitors throughout the study. And during that 2 hours following, they weren't supposed to eat, drink, or exercise or anything else. And in healthy individuals, this test, you would see a spike in their glucose levels right after taking that drink. and then it would slowly come back down to near baseline within those 2-hour time frames. But if a person is diabetic or pre-diabetic or something else is going on, those glucose levels could be higher throughout, and the levels may not drop back to baseline as quickly because they're having an impaired glycemic response. So in these charts, each of them you can see there is on the x-axis the time frame, so kind of an average for each week of the study. And then on the y-axis we have an incremental area under the curve, which is under the glucose curve of these patients' glucose monitoring. And since they were all wearing continuous glucose monitors rather than just collecting blood samples at particular times and having individual glucose values, the researchers had to do this area under the curve analysis in order to compile and compare all the data. And after they did the area under the curve, they compared each individual to their own baseline and averaged this during each week of the study. This statistical analysis was using repeated measures, two-way, ANOVA, and Dunitz. And in general, this area under the curve methodology, you're going to see if the area is higher, that indicates a more impaired glucose response, with the glucose levels being increased or not coming back down to baseline as quickly. So overall, this figure you can see looking at each individual sugar here, or sweetener here, it looks like we have a raise during supplementation in that glucose with saccharin and sucralose, but there's no similar response seen with aspartame, stevia, the glucose vehicle, or the non-supplement control, so both of the control groups. So we can kind of conclude that consumption of saccharin and sucralose can impact the glycemic response in healthy individuals over a short amount of time at least. Now that we have these results, let's take a look at some of the microbiome data that they explained that could provide some explanations. So the first one they decided to look at more closely was sucralose. Because of the 4 sweeteners, it's the one that is most poorly absorbed and most likely to interact with the intestinal microbiome because of that. And as we saw on the previous slide, it was also one of the ones that had the strongest effect on glucose tolerance testing results. So this figure is showing various correlations between features such as stool bacterial abundance and metabolic pathways significantly associated with a change from baseline to the second week of sucralose exposure, but not changed in either of the control groups. There's other parts of this figure, and the paper goes into a lot more detail about a bunch of different plasma metabolites, but we're not going to really talk about those. So in this part of the figure, you can see there's a scale on the bottom left here. On the left, we have purple colors kind of correlating with a lower abundance of that particular pathway or bacteria, and then red indicates a higher abundance. We also have along the right side of this scale down here, you can see the interpretation is correlation with GTT or glucose tolerance testing. So if there's yellow, that means it's positively correlated, and purple is negatively correlated. So looking at this figure in a little more detail, the first 3 rows are showing 3 bacterial species whose baseline abundance correlates with glucose tolerance testing. Specifically, we have Eubacterium, Duria longicatana, and Oscillobacter. We have on the middle 3 rows metabolic pathways that were inversely correlated with glucose tolerance testing, meaning that the activity of these pathways, including arginine biosynthesis, mixed acid fermentation, and the TCA cycle, they all increased in abundance during the supplementation. So you can kind of see that by the color change in the middle 2 here. And then the last 3 rows are showing the abundance of several different purine biosynthesis pathways that were positively associated with glucose tolerance, meaning that their activity was actually decreased during the sucralose supplementation, which you can see in this figure here. So we have some similarly produced charts for the rest of the 4 non-nutritive sweeteners. So this one is showing some of the microbial features and metabolites related to saccharin supplementation. So these are going to be interpreted very similarly to the previous one. You can see we have just a couple things here that were positively associated with glucose tolerance. That would be Prevotella copri and UMP biosynthesis. And then we have a whole lot of bacterial species and pathways that were negatively correlated with glucose tolerance. For example, Bacteroides xylinosolvens. We have a lot of pathways related to glycolysis and glycan degradation that were negatively associated. And you can kind of see here we have a trend of, it started out increased, decreased during the 2 weeks, and then increased again after follow-up. For stevia, there were quite a few different bacteria positively associated with GTT. So you can see a couple of Prevotella species here that their levels decreased during supplementation and then went back to baseline. And on the flip side, Parabacteroides, Lacnospira, Bacteroides coprophilus, a lot of these increased during the supplementation and then went back to baseline. And you can also see that we have a couple of pathways, the TCA cycle and pyruvate metabolism, that pretty much just increased throughout the 4 weeks. And you can see those were positively correlated with glucose tolerance testing. And looking at aspartame, we have some bacteria including Bacteroides fragilis, Bacteroides acidifaciens. These were all positively associated with GTT, so you see a kind of increase during supplementation of some of these. We have some that were negatively correlated, like Bacteroides copricola and some firmicutes. And then several metabolic pathways were also negatively associated, including fatty acid degradation and several biosynthesis pathways. So in all of these non-nutritive sweeteners you can see are affecting the host microbiome and metabolism in pretty different and unique ways. You can see some common patterns throughout where the organism or the pathway is increasing or decreasing in abundance during the 2-week period, but then returning back near baseline values during the follow-up. So the authors are suggesting that this is a good indication that these bacterial species and pathways and functions are directly responding to the presence of the non-nutritive sweeteners. Now, before I go on to what I think are some of the more interesting findings of this paper, I just wanted to mention that the researchers did fecal microbiome transplants from a select few of the human participants in each category into mice in order to kind of observe whether they'd see the similar metabolic results in glucose tolerance testing to the humans that they received the transplant from. So to do this, they took what they called were the top 4 responders and the bottom 3 responders within each of the groups. And what I mean by that is the top 4 responders would be the ones that had the higher area under the curve glucose values during the glucose tolerance testing, and then the bottom responders had lower areas under the curve. So basically the top responders are going to be the people that had the more impaired glycemic responses, and then the bottom responders are going to be the ones that had less impaired glucose responses. So they took the fecal samples from those participants on day 1 and day 21 and transplanted them into the mice, and then they did a lot of the same tests that they did on the human subjects on the mice. So they did glucose tolerance testing and microbiome analysis, and in short, they saw pretty similar findings, that the mice that had top responding fecal microbiomes also had, higher glycemic responses in the impaired glycemic response compared to the ones that had been receiving the bottom responders fecal microbiomes. So this confirmed to them that the individual microbiomes were responsible for causing the varying glycemic responses seen in their human subjects. They were able to kind of attribute causation there. And I would encourage you to read more into this specific part of the paper. It's really interesting, but I don't have tons of time to go into all of the details of it. So on to a bit more about some of the individualized findings that they looked at into this paper, which I think is a really interesting aspect. In this figure, we have graphs again for each of the groups and the two control groups. And these specifically are showing KEGG pathways Bray-Curtis-based dissimilarity to baseline configurations. So basically what that means is they again took those top 4 responders and bottom 3 responders in each group and compared their fecal microbiome features that differentiated them. So they were looking how much did those people's microbiomes and the pathways and the bacteria change from baseline throughout the study. And a higher dissimilarity score is going to be more change. A lower dissimilarity score is less change over the study time. And as you can see, the top 3 charts here showing sucralose, stevia, and aspartame are showing that the top responders had higher dissimilarity throughout the study compared to the bottom responders in purple. On the bottom left, we have saccharin here that is showing dissimilarity kind of converged between the top and bottom responders by the end of the 4 weeks. And most importantly, the dissimilarity was pretty close between the top and bottom responders in the 2 control groups at the bottom right of this figure. So that's kind of showing that the dissimilarity that they see based on treatment in the other ones is significant because it didn't happen in the control groups. So this is the last figure I have. It's the second half of that same figure actually from the previous slide. And it's showing in some more detail some of the exact bacterial abundances and metabolic pathways that differed between the top and bottom responders within each of the groups of supplementation. You can see that there is a key here that shows that the darker the purple, that indicates a higher fold change between baseline and the second week of the non-nutritive sweetener supplementation. Each column is a single individual, and you can see on the left side of each we have our 3 individuals who were our bottom responders and our 4 individuals who were the top responders within the group. So if we take a little bit of a closer look, I'm just going to highlight some key ones to focus on here. Starting with our sucralose group, we have an increase in some pathways related to like glycolysis and the TCA cycle with our top responders, but not with our bottom responders. In the stevia group, the top responders had increases in pathways related to biosynthesis, degradation, and metabolism of some different nucleic acids. So you can see some of those listed here that were higher in the top responders. And then in our aspartame group, the top responders had increases in things like the urea cycle and some of its metabolites, while the bottom responders, interestingly, had increases in an organism called Akkermansia muciniphila, which is actually a probiotic organism associated with metabolic health. So that could be one that's kind of linked to and responsible for protective effects against this non-nutritive sweetener in those bottom responders. In the saccharin group, we have the top responders had increases in caprolactam degradation, which the authors point out is a chemical that's really closely related to saccharin, and it could be a potential mechanism of actually metabolizing and breaking down the saccharin. And then they also had increases in isoleucine biosynthesis, you can see here, compared to the non-responders. And isoleucine biosynthesis can be associated with poorer metabolic health. So as you can see, there's a lot of different things listed here. I would encourage you to maybe dig into some of these. The authors don't really go into any detail about much of any of them, but I think that would be an interesting discussion point to talk about how you think some of these might be related to the glycemic responses. So to get into the discussion about this paper, remember I said the bottom line up front, overall, basically they found that the human gut microbiome can respond to non-nutritive sweeteners and cause variable and individualized metabolic responses in the person consuming them, which has some downstream effects potentially on glucose tolerance. And this is in contrast to the long-held belief that these non-nutritive sweeteners are metabolically inert and can have some pretty broad-reaching implications for human health. But it's kind of also still confusing to interpret. They definitely found that the sugars are causing changes to the oral and gut microbiome, and they found some pretty distinct and specific changes for each sweetener and even between individuals. So I think they definitely identified a lot of stuff that could be used in future research, but there still needs to be a lot more research into these mechanisms behind how these pathways and these organisms in the microbiome can affect the metabolism of these non-nutritive sweeteners. Because since the literature is showing a lot of mixed results, it's really difficult for healthcare providers to know what to tell their patients about these. Is it recommended that they switch to these over sugar, or are these also causing harm in different ways? So we definitely need some more research into all of those mechanisms in order to better understand how they're impacting our health. Some strengths of this paper: I think they did a really great job of including and making sure to screen for people who hadn't consumed those non-nutritive sweeteners in the last 6 months. Their use of the shotgun metagenomic sequencing instead of 16S rRNA allowed them to get a lot more detailed information. I also think that it was really cool that they followed changes over time, like before, during, and after the supplementation, rather just at like 1 or 2 time points. And confirming causation of findings by doing that fecal transplantation into mice was a really cool added benefit of this study. However, some weaknesses, they included only healthy, non-overweight, normoglycemic individuals, so they're missing a huge population of people that actually are taking these supplements. Their use of commercially available sachets rather than purified forms makes comparison between different studies that did use purified forms kind of difficult. And they only studied for a 2-week supplementation period, so I think a longer study could be beneficial. So that brings me to some directions for future studies. Specifically, I think most important, there needs to be more research into the mechanisms behind how these pathways and organisms are affecting the metabolism of these sugars and the microbiome, and how do these pathways exert their effects on the glycemic response. And this is important so that healthcare providers can know how to direct their patients. Are these sugars, non-nutritive sweeteners, good for them as replacements for sugar or not? And again, testing some different patient populations would be helpful as well. So in conclusion, these commonly consumed non-nutritive sweeteners may not be physiologically inert as once believed. They can exert their effects indirectly through impacts to the microbiome, but distinct configurations of the human microbiome are impacted by these non-nutritive sweeteners differently. Here's some questions. I encourage you to pause and take a look at them. I wanted to point out, I've already kind of talked about it on the slides that led up to it, but this third one, talking about different bacteria or pathways that you think might have the impact on the glycemic response and how you think that is. I'd encourage you guys to pick one and do some more research and let us know what you find, because the authors didn't really say much. Here are my references and some images that I used throughout. So I hope you guys enjoyed, and thank you for listening.